So thank you, Andrea. It's great to have you. We're really excited for your seminar. I'm sure a lot of everyone is very familiar with Andrea Ablasser and her work and the impact that she's made in the field of innate immunology. Andrea, I can't look at your CV right now. I had it in front of me just a few minutes ago, but I had to share my slides. But Andrea has done pivotal work in the understanding of how our cells can detect self and non-self pattern recognitions, particularly in the context of nucleic acid sensing. In her postdoctoral work, when she was with Vet Hornung, she made many important discoveries in the understanding of how cGAS and the regulation of cGAMP activates STING. Also made some discoveries in understanding how RIG-I can recognize double-stranded DNA through RNA intermediates. And then starting her own lab at EPFL in Lausanne, she's continued to go on and lead the field in the understanding of regulation of STING and STING inhibitors, and has just made many notable contributions to the field. And she's received also many notable prizes, including the EMBO Award. She's received the Sanofi Pasteur Institute Award, the Coley Award. Over the last few years, the list has just gone on and on. And so it's a real treat to have you here with us, Andrea, today. And we do have a few questions that we like to start the talk off with. And so I was going to ask two questions for you. One, what is I'm Swiss as well. My family is Swiss. And so I was going to ask you what your favorite thing to do, what your top 2 or 3 things are about living in Switzerland. And
okay, so thank you very much for the kind introduction. So what my favorite 2 things are about living in Switzerland?
Yeah. Okay,
so I have to admit that the scenery, the mountains are really wonderful. It's a privilege to live here. And the other thing is that Switzerland is famous for chocolates. So those 2 things are yeah.
And one other question, because our trainees like to always hear from people and how they have got to the place that they are. What would you say was one of the most pivotal moments in your life as it related to you becoming a scientist?
I think it was the experience that I gained when I was a student and did some, I worked for my medical thesis in a research laboratory in Munich. And so the creativity that I experienced by doing experiments and the hands-on experiments I gained, this was really what then, created a lot of excitement and my fascination for research. And so that, kept on growing as I continued my path through my MD thesis. So that is why I then finally turned or deviated a little bit from my initial plan to work as a doctor or practice as a medical doctor, and instead devote all my work towards research.
Great. Well, thank you. Well, we're really excited to have your talk today. So please go ahead and share your slides.
Yeah.
And you're off.
Okay. Can you see my screen in presenter mode or?
Yep. Mm-hmm. Yeah.
Okay, perfect. Okay, so hello everyone. I'm very grateful for the invitation to contribute a talk to this very distinguished lecture series. So as has been mentioned in the introduction, my research focuses on sensing DNA as a dangerous signal through the cGAS-STING pathway. And what originally maybe drove us to explore nucleic acid sensing was actually that it's been known for decades that the recognition of viruses is majorly contributed by the release or the detection of nucleic acids, and that had been established in the 1950s already. And it's been shown already at the time that nucleic acids derived from viruses are very potent triggers of host type 1 interferon responses, which we all know are very essential molecules to restrict viral propagation and spread. But it was less established, despite that this paradigm had been out there for so many years, is how, what pathways would connect the sensing of nucleic acids to the production of type 1 interferons. And what I've found myself, or found at that time very interesting, is that if you think about nucleic acids, they're not pathogen-specific molecules against other pattern recognition ligands. And so I thought this concept of nucleic acids, which are so abundant and so essential for life, can serve as signals for infection. And so we were interested in studying the receptors that would sense those nucleic acids and the signaling cascades that would transduce nucleic acid sensing towards the production of type 1 interferons. And as I said, the key question that goes along with this paradigm is how could nucleic acids be distinguished? So are there specific markers for that are specific for foreign or pathogen-derived nucleic acids that would distinguish them clearly from self-nucleic acids? And as you will see later in the course of my talk, that we know today that receptor signaling cascades and many receptors cannot distinguish based on the structure of molecules, the origin of nucleic acids. But instead, what rather appears to play a crucial role in discriminating self from non-self are, on the one hand, host factors that facilitate the recognition or the activation of these receptors by foreign nucleic acids in the context of viral infection. And on the other hand, we know that there is elaborate safeguard mechanisms that have evolved in parallel to nucleic acid sensors that are essential to maintain homeostasis and prevent accidental recognizing of self and prevent autoreactivity. But clearly there are instances, and I will talk about this as well, in which these safeguards might be overcome and can lead to either immune defenses in the context of cancer, cancer formation or tumor genesis, which can be beneficial, in fact, for the host. But on the other hand, they are also linked to quite severe diseases and immunopathology. So we'll touch upon that in the second part of my talk. Now, what we've been working on in over the past several years, in fact, is how DNA is being sensed inside cells, so from within the cytoplasm. And it turns out that the so-called cGAS-STING pathway, which I think many of you are familiar with, is a key mediator of transducing the presence of DNA within the cytoplasm to the induction of type 1 interferon responses. And this is a very oversimplified slide and summarizing the key components of this pathway. So cGAS binds to double-stranded DNA in a sequence-independent manner, and then it is an enzyme, and this DNA binding triggers its enzymatic activity. It leads to the production of cGAM, which is a unique cyclic dinucleotide molecule that then activates STING. STING is in its resting state an ER localized membrane protein that acts as a dimer. And upon ligand binding, STING oligomerizes and it leaves the ER through the secretory pathway, then moves to the Golgi where it recruits the kinase TDQ1. And then this assembly of an oligomer complex between STING and TDQ1 then allows for the concerted activation of RF3 and NF-kappa B, which then drive de novo transcription of several genes, including type 1 interferons, but also many other innate immune regulators, such as co-stimulatory molecule expression, pro-inflammatory cytokines, chemokines, and other genes, and also sometimes in a cell type specific manner. Now, this pathway is being triggered by DNA, both in the context of pathogen infection, but as we will see later, also in several contexts of sterile damage or sterile injury. Now, concerning pathogen sensing, so this is just a very simplified list and incomplete list of viruses and bacteria that have been associated with recognition through the cGAS-STING pathway. And quite obviously, you find on this list DNA viruses such as herpes simplex virus, which is, so to say, the paradigm trigger of this pathway. But you also find bacteria such as intracellular replicating bacteria like Mycobacterium tuberculosis. But you interestingly also find RNA viruses such as SARS-CoV-2, but also others. We know that cGAS is not being activated by RNA. Instead, these viruses produce collateral damage within the host that then leads to out-of-context DNA accumulation, which then triggers the cGAS-STING pathway indirectly. And in certain cases, this can be protective, and in other cases, this can be detrimental. Now, I just want to quickly point out what I've found a most intriguing development over the past 2 years was that cGAS and STING homologs have been traced back to bacteria. And in fact, we know that the same pathway components are actually very important for protecting bacteria from phage infection. I think this points towards a fascinating conservation of this pathway throughout billions of years of evolution. Now, what concerns cGAS activity, there have been through both biochemical and structural studies over the past years been gained very detailed insight into the functioning of this enzyme. And so on the left-hand side, you can see a very simplified schematic of the cGAS protein. So it has an N-terminal unstructured domain, which is positively charged, and that facilitates interaction with the negative charged backbone of double-stranded DNA. And then the human catalytic domain is composed of 3 DNA binding sites, which are called A site, B site, and C site. And upon DNA binding, cGAS undergoes a conformational change, which liberates the enzymatic pocket so that it can be can bind ATP, GTP, which are then the substrates for the subsequent synthesis of cGAM. But what's also important to note is that cGAS works as a dimer. So the minimal active unit of cGAS is a dimer in which 2 cGAS polymers are sandwiching 2 strands of double-stranded DNA, which cover the A side and the B side respectively. And then in addition, the C side can also bind to DNA, but it's not important for dimerization. But instead, what the C side have been shown is that it helps to oligomerize cGAS multimers. And so in fact, we know that even so the minimal active unit is a dimer, in vivo dimerization might not be sufficient to trigger cellular responses. Instead, this requires oligomerization of cGAS, either forming those ladder-like assemblies or even phase-separated condensates. So this oligomerization on long strands of naked DNA appears to be a key determinant for cGAS activity. And maybe that also serves already as a first filter to filter out self-DNA, which is, or DNA, which is long stretches of naked DNA, which are typically not found within the host cytoplasm of a healthy young cell. Now, however, if you think a little bit about this pathway, then you might maybe note that there is an enzyme which, in a ligand unspecific fashion, can produce a very powerful second messenger, which then activates a very potent innate immune response. And so I think this pathway seems to be really perfectly positioned to sense broad classes of very different pathogens. But at the very same time, it also is tempting that this pathway might also, sense self-DNA. And of course, that then implicates that there might be tight regulatory mechanisms that prevent self-DNA from activating this pathway. And so we have, over the years developed a strong interest in understanding how this pathway operates at a cellular but also biochemical level, and moreover, how if this pathway becomes deregulated, how using this knowledge we can maybe devise new therapeutic paradigms on how to intervene into certain inflammation-associated conditions. And so what I would talk or cover today within this talk is, on the one hand, mechanisms that restrict cGAS on self-DNA. And then for the second part of my talk, I will address how targeting this pathway could, might be beneficial in the context of immunopathology, and specifically in the context of COVID-19. So in the early days after its discovery, there emerged largely two paradigms on how nucleic acid sensing or DNA sensing through cGAS might be regulated. And you could place these mechanisms in, or there was largely these two major categories, which is on one hand, degradation of self-DNA in places where cGAS localizes. And on the other hand, physical separation, so that membrane compartments, such as, for instance, the mitochondria or the nuclear membrane, might limit the access, the physical access of cytosolic activatable cGAS with DNA. And we know today that, for instance, deficiency in nucleases that degrade self-DNA, such as DNase2 in the lysosomes or TREX1 in cytoplasm, can trigger a cGAS-dependent type 1 interferon response. And for instance, this is an example showing you at the cellular level that TREX1's deficiency, so a nuclease that's essential for degrading DNA in the cytosol, deficiency triggers type 1 interferon responses, which are completely abrogated by a concurrent knock out of cGAS. Now, mutations within this gene are linked to a very severe human syndrome called Akadi-Gutierrez syndrome. And it has also been shown by the laboratory of James Chen and Dan Stetson that depletion of cGAS completely rescues the phenotype of TREX1 deficient mice, which suffer from a very severe lethal autoimmune syndrome, and that can be completely rescued by the deletion of cGAS. Now, with regards to physical separation, there have been a number of studies that had shown that DNA damage responses can trigger cGAS activity by cGAS sensing damaged DNA in the cytoplasm. And that's an example of our own work in which we showed that inside senescent cells, cytosolic chromatin fragments, which are characteristic features of senescent cells, can be sensed by cGAS to trigger an inflammatory response within these cells. So, but both neither degradation nor physical separation could explain how cGAS might be regulated in dividing cells during mitosis. And what you're looking here, and this slide is a video in which cGAS is shown in green, and upon nuclear envelope breakdown, cGAS binds to chromatin. It's actually carried into the nucleus. And we know today that many cells, both dividing and non-dividing cells, contain a considerable fraction of their cGAS pool within the nucleus. And maybe that seems at first a little bit counterintuitive. So the question that emerged from these studies that showed that cGAS locates inside the nucleus, where it's tightly tethered to chromatin, excuse me, was how cGAS might be regulated on chromatin. And sorry. And so there we had over the past years conducted several studies in our laboratory that led us to formulate the hypothesis that the nucleosome core particle itself, which is a key feature of intact genomic DNA, might serve as a critical regulator of cGAS inside the nuclear space. And I would just briefly highlight a few of the key experiments that led us and others to make this hypothesis. And probably one of the key experiments that we and others performed was that we observed was when we reconstituted in vitro cGAS activity by DNA. And what we found is that cGAS in the presence can not only not become activated in the presence of nucleosomes, but a bit surprisingly is that if one would titrate nucleosomes into a productive cGAS reaction, that cGAM synthesis can be effectively inhibited, and that's what you're looking at here. So adding more nucleosomes or chromatin fibers would inhibit the cGAM output of this in vitro reaction. What was also notable is that this inhibitory effect could at least partially also be recapitulated by using H2A/H2B dimers, so subunits of the nucleosome core particle that lag DNA. And that was a very interesting observation because it led us to the idea that potentially there might be important protein interactions that are mediated by the H2A dimers, and that could mediate this inhibitory effect that we observed. So at that time, we then initiated studies in which we reconstituted cGAS binding to nucleosomes in vitro, and we obtained cryo-EM structure of human cGAS bound to the nucleosome. And before I start the movie, what you can already see here is two nucleosomes that are somehow cross-bridged by two cGAS polymers. And so what is interesting now, what you can appreciate maybe, is that cGAS appears to utilize both DNA, its canonical DNA interactions to bind to the nucleosome, which is my binding to DNA, so to bind to the nucleosome. But then it seems to also utilize a quite extensive protein binding surface to bind to the surface of the nucleosome, as shown here. So now we could further refine the structure to 3.1 angstrom final resolution of this proximal cGAS or cGAS binding to this proximal nucleosome. And if you zoom in, you can now really appreciate the kind of extensive interaction that cGAS has with the nucleosome surface. Now, if you zoom in, it seems that there are three loops, 1, 2, and 3, that largely contribute to the cGAS tethering to the nucleosomes. And these are a charged interaction, and it seems that the major contribution to this charged interactions are originating from two arginine residues on the cGAS side, arginine 255 and arginine 236, and on several charged amino acids on the H2A/H2B dimers that are known to form a very prominent spot on the nucleosome, which is the so-called acidic patch. Now, this interaction is further facilitated by canonical interactions of the cGAS-B side. So these are residues that contribute to DNA binding through phospho or DNA backbone interaction, as shown here. Now, using biochemical assays, we confirmed that mutating either this arginine residue or this arginine residue to alanine led to a complete abrogation of cGAS complex formation with the nucleosome core particle, as you can see here by this EMSA shift assay. And moreover, those mutants were no longer inhibited by chromatin, so indicating that the tethering of cGAS to this acidic patch surface appears to be a major restricting factor in limiting cGAS enzymatic activity. Now, and indeed within living cells, we could confirm this phenotype in that either mutating arginine 255 or arginine 236 led to cGAS untethering from its very tight interaction with chromatin. It led to spontaneous production of cGAMP, and it also induced spontaneous type 1 interferon induction. So this led us then to propose the following mechanism. So as a minimal active cGAS DNA complex requires dimerization as shown previously, and cGAS cannot undergo or cannot adapt this complex once it's bound by the nucleosome. And largely because the B site is used to bind cGAS to the nucleosome surface and also to interact with nucleosomal DNA. The A site, even though it's partially solvent accessible, is sterically blocked. So long stretches of double-stranded DNA would not could not be bound in that configuration. And obviously also the dimerization of a second cGAS polymer would be inhibited by the nucleosome. And so we think that this explains why once cGAS is bound to the nucleosome, it cannot productively produce cGAMP and neither interact with a double-stranded DNA. Since this interaction or the affinity for the nucleosome is preferred over the affinity of double-stranded DNA, this also explains why cGAS cannot undergo activation inside the nucleus. So I should mention here that very similar or identical structures and similar reports were at the same time reported by the Hopfner group, the Pinweli group, the Funabiki group, and also the McGinty group. Now, what's interesting is if you trace back cGAS in evolution, we find that these arginine residues are highly conserved in cGAS homologs that use DNA to drive enzymatic activity, but they seem to be not conserved in cGAS, for instance, from sea anemone, which we know is not driven by DNA sensing. So perhaps that points to an interesting co-evolution of DNA binding and nucleosome binding in that once cGAS developed the capability to use double-stranded DNA as a driver of activity, and at the same time invented a very powerful mechanism to restrict its activity once it's confronted with self-DNA, which in that case is being identified or being read out by the presence of nucleosomes. And so now to kind of adapt or further develop this, the concept on how cGAS is being regulated. So I showed you before that DNases, so degradation and physical separation, are key mechanisms to restrict cGAS inside the cytosol. And here the presence of DNA is sufficient to trigger cGAS activation, which can be induced by infection, by damage, or by certain types of, especially genotoxic therapies. But nuclear regulation might work differently. Here, the activation threshold of cGAS is relatively high because it's steadily inhibited either by nucleosomes or also by limited DNA accessibility, which is imparted by chromatin architectural proteins. But now you can imagine that maybe that cGAS might overcome this inhibition and certainly also be activated inside the nucleus, which might be relevant in sensing infection, in sensing damage, and potentially could also be exploited therapeutically. I will switch now gears and talk about the cGAS-STING pathway as a therapeutic target for inflammatory diseases. cGAS is involved or have been associated with a huge variety of distinct human pathologies, most obviously viral infection, which is often protective, and then also in both natural and therapy-induced anti-tumor immunity. In fact, we know that cGAS or STING deficient mice in several preclinical models of cancer are highly susceptible towards tumor growth and also lethal outcome. But what also emerged is that this pathway is involved in the pathogenesis of a variety of inflammation-associated conditions. And here I depicted a few examples, especially it seems that there is quite association with neurodegeneration, such as Parkinson's disease, ALS, FTD, Huntington's disease. You also find association with cardiovascular disease, also more classical autoimmune syndromes such as lupus, arthritis, or vasculitis, and also certain types of largely inflammation-triggered carcinogenesis or metastasis have been linked to this pathway. And at the origins of all this non-infectious or sterile-triggered activation of the pathway lies the sensing capability of cGAS of self-DNA. And there are many different sources of self-DNA that can fuel into this pathway and promote activation in these contexts. For instance, extracellular DNA, largely necrotic or apoptotic cells, can trigger this pathway, for instance, in cardiovascular disease. Mitochondrial DNA leakage had been a prime trigger of cGAS in certain neurodegenerative diseases. Then DNA damage and broken mitochondria are obvious targets for cGAS and the link to cancer. And also endogenous retinal element are upregulated in certain contexts, such as aging. And so at the end, what appears is that out-of-context DNA is a critical hallmark of many non-communicable diseases that can, through the cGAS-STING pathway, trigger either an acute inflammatory reaction or a more chronic inflammatory reaction, which in both cases might, independently of their original trigger of the disease, promote the disease and drive pathogenesis. And so with this in mind, what we hypothesize is that targeting this secondary response towards out-of-context DNA might potentially be a useful path to intervening into chronic or acute inflammation-driven disorders. And so some time ago, we discovered small molecule inhibitors of STING that are very specific in targeting STING, and they do so via a very specific mechanism. So very briefly, I told you before that STING works as an oligomer at the Golgi, where it recruits the kinase TBK1. And this oligomerization appears to require a post-translational modification on STING, which is the addition of a palmitate. So it necessitates palmitulation. And what the compounds that we identified do is that they irreversibly target cysteine residues, in particular the cysteine residue 91, which is at the interface of a transmembrane and the cytosolic loop of STING. And in that fashion, they inhibit palmitulation. Excuse me, oligomerization of STING, and that impedes the recruitment of TBK1. And as to the best of my knowledge, these compounds are thus far major compounds, the only compounds that can effectively target STING in vivo contexts. And we've demonstrated this as a proof of concept using JAK1 knockout mice and treatment with this indole-based H151 compound for 1 week led to significantly reduced type 1 interferon responses in vivo in many different organs, as you can see here. And this is quite a remarkable effect considering the profound type 1 interferon response that you see here. So with these compounds at hand, we could now test the involvement of STING in many different conditions. And what I will talk about now for the last part is on the control of the cGAS-STING pathway and its involvement in the pathogenesis of COVID-19. So I guess everyone here is familiar with the immune response trajectories in very severe cases of SARS-CoV-2 infection, in which it was early on shown that severe disease is associated with a hyperactivation of the innate and then also the adaptive immune response. And what was, from our point of view, very interesting with this regards is that type 1 interferons appear to play a dual role in COVID-19 pathogenesis. And it's very well known that type 1 interferons, if they're produced early, they're essential in rapid viral clearance, and deficiency in type 1 interferons or antibodies that target interferons can promote severe disease. But What had also been shown later on is by following trajectories of immune correlates that would indicate severe disease, was that patients with severe disease often showed a delayed type 1 interferon response or an exaggerated or persistent type 1 interferon response, which would trigger severe inflammation and disease. And that had also been recapitulated by the recombinant treatment with interferon, with recombinant interferon. This is from a retrospective study, which was shown that if given early in the course of disease with mild symptoms, type 1 interferons are decreasing mortality, but later in the course of the infection, they might even increase mortality. And so these opposing functions of type 1 interferon, especially the late detrimental response, seemed interesting to us, and we were wondering whether there might be any connections of the cGAS-STING pathway to contribute to this late interferon response, and eventually also to the observed immunopathology. And to study this question, we took advantage of skin biopsies of severely diseased COVID patients. So they, I'm sorry, I just need to. As you know, COVID, even so it manifests largely in the lung. It also has extra pulmonary manifestations, and among them is also the skin. And these extra pulmonary skin manifestations, they are quite heterogeneous. So they come as macular rushes or purpura necrotic lesions. And we collected those lesions and profiled them in comparison to other skin diseases. And what we found was that SARS-CoV-2 lesions, that these biopsies, that they clustered in close proximity to cutaneous lupus samples. And if you zoom in the genes, you find that many of the genes, based on this clustering, fall into either the category of type 1 interferons or interferon-induced genes. What was interesting that lupus and COVID patients, they showed a very distinct profile with regards to macrophage activation markers. In that sense that COVID patients showed upregulation of macrophage markers such as key cytokines, key differentiation markers, and key receptors of macrophages, and these were not present in the skin lesions of lupus lesions, which is consistent with the paradigm that lupus is largely triggered by dendritic cells, and which also befitted really similar first studies on showing the major contribution of macrophages and myeloid compartment in driving hyperactivation of innate immune response genes in the context of COVID-19. So we were then wondering whether indeed macrophages are the origins of the type 1 interferon response that we detected. And by staining those lesions, we found that interferon beta really indeed enriches in CD163 positive cells, which are macrophages. Interestingly, however, macrophages were not, as you can also see here, the only participants to that response. And we also observed that there is marked interferon positivity in cells that stain positive for CD31, which is an endothelial marker. And throughout the 10 lesions that we biopsied, you can see that there is a varying contributions of both endothelial cells and macrophages. Now, what these biopsies also revealed is that endothelial cells in COVID showed this marked signs of endotheliopathy, which here is shown by cell swelling, or also increased cell death, as shown here by caspase-3 positivity. And interestingly, the amount of caspase positive cells correlated with the type 1 interferon intensity. suggesting a possible link between those two events. And so we were wondering whether maybe dying cells could act as a trigger of this type 1 interferon response. And so indeed, when we stained again macrophages, which are interferon positive for DNA, we found that those contained very prominent signals of DNA within their cytoplasm. And we also showed that they contain caspase 3 positive fragments in their cytoplasms, indicating that these phagocytes, which are interferon positive, had engulfed dying cells. Now, would there also be direct evidence of the cGAS-STING pathway? And so we addressed this by measuring cGAMP in the skin, which is the most specific marker of cGAMP activity. And you can see here in 7 out of 10 patients, there was an elevation of cGAMP production. Moreover, we detected phospho-STING, which is a very specific biomarker of activated STING, both in macrophages, as shown here, but also in endothelial cells. And so that shows that in addition to type 1 interferons, there are biomarkers that indicate that the cGAS-STING pathway might be active. But was it also causally linked? So what we did next is we used these STING inhibitors and cultured skin biopsies in vitro, and then measured ISGs as a readout for activity. And what we found is that H151 inhibited this interferon response, as shown here by several independent ISGs. And so that indicated that there is not only a correlation between type 1 interferon production, cGAS-STING pathway activity, but in fact that there might be a causal link. So that was not specific to the skin. We also obtained post-mortem lung samples. And what we've observed is that in early diffuse alveolar damage, so lungs that showed sign of early diffuse alveolar damage, we observed that phospho-STING was activated in macrophages, but not in samples that were associated with already signs of regeneration and fibrosis, and which are known as late diffuse alveolar damage signs. So this was specific to this early stage. And interestingly enough, also only early stage DEDD showed upregulation of type 1 interferon genes, as shown here by MX1 staining. So again, indicating that not only in the skin, but also in the lung, there seems to be a correlation between activation of the cGAS-STING pathway and heightened type 1 interferon responses. And again, both in the skin and also in the post-mortem lung, we observed this endothelial cell engagement. So the question that we now had is, it seems the macrophages are activated by dying cells, but we were also intrigued by this effect on endothelial cells, given that endotheliopathy is really a hallmark feature of COVID-19. And so what we were then wondering, what might drive this type 1 interferon response in endothelial cells? And to do so, we needed to recapitulate the infection of endothelial cells in vitro, and it is known that as monoculture, endothelial cells cannot be infected with SARS-CoV-2, largely because they lack ACE2 expression. And so to circumvent this problem and be able to transfect or be able to obtain endothelial cell infection, we turn to a lung-on-tube model. And within this model, there is an epithelial layer of epithelial cells, which is exposed to an airway interface. And on the vascular sides, there is endothelium. We can also be where we can add macrophages to reconstitute also macrophages on this vascular side, also on the airway interface. And also this vascular channel allows for the perfusion of drugs such as H151, and that allowed us to really interrogate the effect of SARS-CoV-2 infection in a system that mimics the in vivo situation. And what we found first is, again, what you're looking here at is the airway interface, so the epithelial side, and after infection, we observed little type 1 interferon responses in the epithelial cells, Here you see some macrophages outlined in green, and it's very consistent with with many other studies that have shown that SARS-CoV-2 is very efficient in curbing interferon responses. They have very prominent mechanism in antagonizing this response. So there's only little productive interferon production. However, on the vascular side, this is what you're looking here now. On the vascular side, we observe prominent type 1 interferon responses, both in the macrophages, but also in endothelial cells. And intriguingly, this was entirely plummeted after the addition of H151 to the vascular side. Importantly, there was no major difference or no significant difference in SARS-CoV-2 replication in the epithelial or in the endothelial side, meaning that this inhibitor not only simply affected this response by restricting infection. Another notable observation was that not only interferon is being induced by SARS-CoV-2, but also that what we observed in vivo, that these cells undergo cell death, as shown here by caspase-3 staining. Interestingly, this effect was also completely dependent on STING. We observed, again, phospho-STING activation, indicating that STING is really activated in these cells. And moreover, by looking at many different genes, we observed that in endothelial cells, type 1 interferon genes are effectively reduced by STING inhibition, but also endothelial markers such as tissue factor, which has also been linked to this endotheliopathy. There is no major impact on the epithelial side, which would also just produce very marginal type 1 interference. Now, what would be the driver of interferon in these endothelial cells? Now, we did unbiased mass spectrometry analysis of the infected endothelial cells from this lung-on-chip model, and what we identified was that many proteins were upregulated that were somehow linked to the mitochondria. And when we did transmission electron microscopy studies, we observed that in these SARS-CoV-2 exposed endothelial cells, we observed this disruption of the mitochondria, as can show here. And interestingly enough, this was also evident from in vivo biopsies of COVID-19 patients, that there is a disruption of the mitochondria, and that had also been independently been shown by other proteomic studies, for instance, by the Peterman group. So this is just a quantification demonstrating that SARS-CoV-2 of infection of the lung-on-chip resulted in significantly changes in mitochondria, which appeared more swollen and disrupted. That, of course, pointed towards mitochondrial DNA being the trigger for cGAS in the endothelial cells. And to test this, we depleted mitochondrial DNA using DDC pretreatment, as shown here, and that led to a significant reduction of type 1 interferon in these endothelial cells, so indicating that indeed mitochondrial DNA is a driver of this response. Moreover, we used an inhibitor of a voltage-gated anion channel that had been shown to be a gateway for mitochondrial DNA release, and which is called VBIT4. And similar to DDC treatment with VBIT4 inhibited type 1 interferon induction in this model. So we conclude that it appears that type 1 interferon or cGAS promotes type 1 interferon induction and endothelial cell dysfunction in this lung-on-chip model, which is kind of a very solid mimic of the in vivo situation. And so finally, we asked whether STING also plays a role in vivo and whether we could target it with the STING inhibitors. And so to do so, we first tested whether STING plays a role in this model, and therefore we prophylactically treated human ACE2 expressing mice under the control of the K18 promoter, which is a very well-established model of SARS-CoV-2 in mice. We treated them with a STING inhibitor 1 day prior to infection, and then at day 6 after infection, harvested lung and looked at many different parameters. I should mention that this model of SARS-CoV-2 infection is a very severe model, and typically after 6 days, most of the mice need to be sacrificed because of the severe degree of disease. So what you're looking at is histopathology of the lung, and as you can see here, that there is less infiltrate in the H151 treated group. We also observed by tunnel staining less accumulation of dead cells in the inhibitor treated group. And when we checked cytokine levels, we observed that across many different cytokines, interferon, pro-inflammatory genes, chemokines, lung integrity markers, STING inhibitor had a significant effect. Notably, some of the interferon effects are only reduced, whereas others appear to be completely abrogated. What was interesting was that this was at day 6 after the infection, so at a relatively late time frame. But when we checked at day 3 after the infection, which is around the peak of viral replication, the STING inhibitor had no effect on interferon response genes and neither IL-6. And so that indicates that the early wave of interferon and inflammatory cytokine induction is independently of STING, and only the late response is mediated by STING. We verified that neither in the lung or the spread to secondary organs such as the brain is affected by the STING inhibitor. And finally, also at day 6 after the infection, we measured the ongoing interferon response by looking at phosphostat-1 signaling, which was severely reduced in STING inhibitor treated mice compared to vehicle control. And also NF-kappa B responses, as shown here by phospho-P65, have been reduced. Then finally, we also looked at overall weight changes, and there seemed to be a significant improvement of the SARS-CoV-2 infected mice that had been treated with H1.1. Now, this was prophylactically, and to translate this to a clinical relevant situation, we also used STING inhibitor treatment at day 2 after infection when the peak viral replication occurs. And very similar to before, we observed significant reduction of lung pathology at day 6, reduction of weight loss that might even be more efficient than the prophylactic treatment, and also eventually rescue of survival and survival experiments. So that led us then to the summary that upon SARS-CoV-2 infection and replication in the epithelial cells, endothelial cells are triggering a wave of cGAS-STING responses, which is triggered by the mitochondrial damage that the virus induces and triggers type 1 interferon responses and cell death. And eventually also macrophages are being recruited to the cGAS-STING dependent response, which sends DNA from dying cells and produce type 1 interference, which then secondary full inflammatory responses. Now, I think there are important implications from the study. And first of all, it seems that the opposing functions of type 1 interferon can now mechanistically be discerned. It seems that the early wave of type 1 interferons, which are largely protective and correlating with mild disease, are not dependent on cGAS-STING signaling, but are exclusively being triggered by RNA sensing toll-like receptors or RIG-I-like helicases. Instead, the cGAS-STING pathway only impacts later responses, later type 1 interferon responses, which develop secondary to the infection as a response to tissue pathology. What I find very interesting is that there have been reports on mutations, single point mutations in STING that derives STING homologs in bats, which or render STING less capable of making type 1 interference, and that might be one of many other reasons why bats might sustain highly pathological coronavirus viruses such as SARS-CoV-2. And of course, also maybe that points towards that STING or cGAS inhibitory compounds might be promising avenues for select intervention into detrimental interferon responses in in COVID-19, but also other detrimental viral infection models. Okay, with this, I'd like to acknowledge the people that have contributed to the work that I've been showing to you. So first of all, for the nucleosome structural paper and project, I'd like to acknowledge Alexiano, who was a postdoc, and Selene, who was a student in my lab, and this was a collaborative project with the lab of Nicolas Thomain in Basel. And the second part of my talk have been carried out by Fatih in my lab, together with Michel Chidi at the University Hospital of Lausanne and Vivet at EPFL. Thank you also to the funding that supported the research that I've shown, and I'll be happy to answer any questions. Thank you.
Great. Thank you so much, Andrea. That was just a wonderful talk, and so many different functions that that cGAS and STING are playing a role in, and just the amazing translational work that's going on in your lab. It's really exciting. I'm going to share the screen and let people see how they can get in touch with you, because we'd love to have people ask you questions. And so the way that we enable you to ask questions is to reach out over Twitter. And so if you search for the account Global IMMUNOTALKs, and then you'll find a tweet that says ask questions for Dr. Andrea Ablasser here. And it's at ImmunoSpeaker. So go ahead and you can hashtag Global Immuno as well. But she will reach out over the next couple of days and answer any questions that you might have. And I just want to let you know who's coming up. Next week we have Michelle Linterman. And then after that, Vet Horgan and Kristen Hoquist. So please keep joining us each Wednesday at 9 a.m. Pacific Standard Time, 12 noon Eastern Standard Time, 5 p.m. GMT time. And thank you again, and have a great day, evening, morning. All right. Thanks, everybody. Bye.